So we will jump right into our first deep dive section, which will focus on gene therapy. We're very happy to welcome our moderator today, Adam Shaywitz, and I will bring him up on the screen here. We're very happy. Thank you, Dr. Adam Shaywitz. Adam Shaywitz is Chief Medical Officer at BridgeBio Gene Therapy and an incredible Cure Sanfilippo Foundation board member. We're very lucky to have you. Take it away, Adam. I'll bring up your slides right now.
Great. Thanks, Glenn. Thank you for that really powerful and inspiring introduction. And I continue to be blown away by the work that you and Cara and the entire foundation is doing with the support of the community. I first met Glenn and Cara probably about 10 years ago when I was at Byrne working on developing an enzyme replacement for Sanfilippo type B, and was just so impressed with their thoughtfulness and dedication, and was honored when they invited me to become a board member about 5 years ago, and continue to really be inspired by the work here and driven by this real need. So we can go to the next slide. You saw my conflict of interest slide. I work at BridgeBio and still have some stock at BioMarin. I want to be brief to respect what Glenn was just talking about. We need to move quickly, but I did just want to briefly mention, it's a very, very exciting time in gene therapy. My day job, I work developing gene therapies for rare disease. I'm keenly aware of how much excitement there has been, and I'm sure all of you in the audience have heard the power of gene therapy in the lay press, maybe even in the biotech or scientific press. We've seen the tremendous potential of drugs like Zolgensma for spinal muscular atrophy. We see headlines about curative, magic bullet, transformational. But on the flip side, I think there's a balance. You can go to the next slide. We do also realize and are growing to appreciate more and more that there are also, some things that we need to be careful about with gene therapy. There are concerns around durability. How long will it last? Will my child be able to get redosed if it doesn't last, or if they don't have as good an effect as they'd like? And I know Rafael is going to talk about this a little bit later. There are immune reactions that we know happen with many types of gene therapy. These immune reactions can run the range of just lab changes to really serious adverse effects and really serious complications. And I know Dr. Byrne is going to talk about this. So I think you can go to the next slide. Gene therapy is really all of these things. It's a type of therapy that has tremendous potential. We've seen that, but we also need to be aware of the real side effects and the burdens. And I know Cara is going to talk about that during her last presentation. And so it's really important, and I'm probably preaching to the choir here, but it's really important to really try to understand what type of gene therapy you're considering. There are many different types of gene therapies. There are different ways that gene therapy can be delivered. Glenn just mentioned there are different, even within Sanfilippo, there are more severe forms and more mild forms and even intermediate forms, as Glenn and the foundation are uncovering. So there are so many individual elements of considering how gene therapy might impact your child, from the underlying disease to the therapy to how it's delivered, that it's very hard to kind of say that there's a one size fits all for the risk benefit. The risk benefit is very individual. But we can go to the next slide. Just to provide some context before we go into the talks, it's important to understand the different types of gene therapies and the different risks that are associated with the different types of gene therapies. For example, today we're going to focus on AAV gene therapy, or most of the talks will be on AAV gene therapy today. And there are two types of gene therapy. There's systemically administered gene therapy, where we just take a gene therapy and inject it right into the body. We could inject it into an organ. We can inject it into the bloodstream. We can inject it into other bodily fluids like cerebral spinal fluid. Usually that's done with AAV gene therapy. There's also another type of gene therapy where we take the cells, a child cells out of their body, we put them in a flask or a petri dish, and then we add the gene therapy outside the body. Their cells take up the gene therapy, and those cells are then reinfused. So for example, the recently improved Libmeldi for metachromatic leukodystrophy, another very rare, really debilitating disease, that is an example of ex vivo gene therapy with a different virus, a lentivirus, whereas Zolgensma for spinal muscular atrophy is an example of systemically administered AAV gene therapy that's injected into a vein, typically in the arm. Next slide. And there's also different ways that we can use gene therapy. We can simply add a working copy of a piece of DNA or gene that isn't working. This is gene augmentation. So we're not worried about correcting what's in the DNA. We're just worried about restoring activity that isn't there. So for example, Zolgensma, they're not correcting the underlying genetic mutation, but they're just adding a working copy of a gene that will help replace the missing or defective protein. For other types of newly emerging gene therapies, they're using gene editing, which is a newly evolving technique where they actually can take new molecular machines and change the DNA to correct the underlying mutation. So you're not adding something, you're actually changing and correcting, reverting the mutation in the genetic code to something that is now producing a working copy of the missing or defective protein. So that's a new technology. Each of these technologies have their own risks and benefits. So it's really important when we think about risk and benefits, immune reactions, each of these types of situations, AV versus lentivirus, systemic versus local delivery, gene augmentation versus gene editing, they each have their own risks and associated potential benefits. So with that, I will turn over to our speakers. We're very lucky to have a really expert panel here with significant experience in gene therapy in clinical and preclinical development. Our first speaker is Dr. Barry Byrne. Dr. Byrne is a clinician scientist, a pediatric cardiologist by training, who is studying a variety of rare diseases with the goal of developing therapies for inherited muscle disease. His group has made significant contributions to the understanding and treatment of Pompe disease, a disease characterized by abnormal accumulation of a sugar called glycogen in the muscle. His team has also been developing new therapies using AAV technology to restore muscle function in Duchenne muscular dystrophy, Friedreich's ataxia, Pompe, and other inherited neuromuscular diseases. His group at the Powell Center at the University of Florida has also established a series of new methods for large-scale AAV clinical manufacturing, and his work is supported by several NIH industry and foundation awards. He is the Associate Chair of Pediatrics and Director of the University of Florida Powell Center and Child Health Research Institute. And in addition to all of his academic work, he serves as the Chief Medical Advisor of the Muscular Dystrophy Association. So with that, I will turn over to Dr. Byrne.
Thanks very much for the kind introduction. I hope you can hear me all right. Okay, perfect. So if we can advance to the first slide. Thank you for the kind invitation to participate in the meeting. I want to give you some practical considerations about the immunology associated with AAV gene therapy. This has been one of our strong interests. For the past 10 years, we've been developing new methods which will allow inclusion of a greater number of study participants, or in the case of commercial therapy, access to commercial therapy. And also, because of the prospect for early treatment when there is an opportunity for newborn screening, we want to make sure these therapies are durable throughout the lifespan. So we're focusing also very strongly on re-administration of gene therapy vectors which was previously thought not to be possible, but I hope I will convince you by the end of the discussion today. We have several strategies to address that need. So next slide, please. So when we think about at least systemic administration of AAV, there are two important parts of the immune response. One is the part of immunity that's ready at any moment when there is an invading pathogen, and that's known as the innate immunity. That can take over the response within minutes to hours after exposure. And in fact, what's very unprecedented about systemic use of gene therapy agents is we never really are exposed to such a large amount of any viral protein or viral pathogen instantaneously, like when administering a systemic AAV gene therapy. So it's quite unusual for the immune system to be confronted with such a challenge. And this is where we see some of the principal safety events, and I'll discuss some of what we've done to try to address that. And then acquired immunity begins in days to weeks following the administration. These are the antibody and cellular immunity responses that I'm going to focus on, particularly as part of inclusion or exclusion criteria for gene therapy studies or commercial gene therapy. So these two arms of the immune system work together to cover really all the possibilities to keep us free from harm from invading pathogens. And after all, a gene therapy vector is a re-engineered virus, which whether it's the lentivirus that was just talked about, which can be also administered in vivo, is commonly used ex vivo outside of the body. But with systemic administration of AAV vectors, this is where the innate immune system plays a big role. So you can go on to the next slide. I wanted to point out that for those that want to learn more about this offline, when they have a chance to look at this plain language summary that we published last year with colleagues at Pfizer. And the goals of the plain language summary are to really take some of the more complex principles of immunology and address them in a straightforward manner that's understandable by everyone, to understand the role of immunity in protecting us from infection and how it impacts the opportunity for treatment, like with a gene therapy product. So the title of the summary is here, and I'll go into a little more depth with the examples from that plain language summary, which I hopefully will be useful to the community. Next slide, please. The purpose of this plain language summary was to help explain why antibodies against a virus can prevent its use therapeutically, and also explains the basis for which, after an initial exposure, there is a very strong antibody response, which we believe in some cases is not just a limitation for access to these products later, but also is the principal driver of the safety events that have been seen in AAV gene therapy studies, because it really wasn't anticipated that the human responses would be fundamentally different from those in the preclinical models. Certainly, we know that rodents tolerate many of these issues very easily, and that was not a concern based on the rodent preclinical studies. But what was a surprise is we always felt like primates were a good model for the expected responses in humans. But primates don't have the same hygienic lifestyle that humans do, and in fact, they've attenuated many of their innate and early adaptive responses to be able to ward off or coexist essentially with many chronic viruses that infect primates. So the next slide, I'll go into a little more detail. The way these antibodies are tested for is important to understand, so that when eligibility is being considered for a study or commercial product, there are two principal ways the antibodies are detected in the blood. So these are both based on blood tests from the participant. And unfortunately, we don't have a universal test to use to detect AAV antibodies. The way this has been handled with the regulatory agencies is each sponsor develops an assay that's specific to their product. So these are called companion diagnostic tests. And there are either two types, one in which the binding of the antibody to an AAV antigen or vector is done in a system which can detect the antibody binding with a second antibody. This is known as an enzyme-linked immunoabsorption assay, or ELISA. And the ELISA method we feel is very sensitive at the lower limit of detection, which is where these pre-existing antibodies, normally the level is relevant for measuring that participation in a study. The other type of assay is called a neutralizing antibody assay. And in this case, the AAV is used to infect cells in culture, and the ability of the antibody to prevent that infection is studied using the neutralizing assay. I think predominant number of studies are using the binding assay. The next slide. And so we ask, well, one is, are these assays accurate, and what do they reflect in terms of the real world experience? So we've studied about 113 patients now over a 5-year period to look at pre-existing seropositive status, so for AAV9 in this case. And we know that, as you can see, over year, the average antibody values don't change much. There are fewer patients that we've had at 4.5 and 5 years. But in general, the antibodies, particularly if they're post-exposure, are significantly higher than this and very long-lasting. So one notion that we wanted to be sure to be clear about is, if there's not considered how to prevent the antibodies from forming in the first place, they'll be persistent and very long-lived. And that's because the cells that make the antibodies are called long-lived plasma cells, and those plasma cells can continue to make antibodies even if they're removed transiently by an antibody lowering method. And so that's why we want to focus on both the plasma cells and the antibodies in considering ways to prevent responses to AV. So in the next slide, another consideration that comes up if we do have newborn screening is, do the antibodies of the baby reflect that of the mother, or just of the exposures the baby might have had in utero? We know that the IgG molecule, which is the one most commonly formed after exposure, does cross the placental barrier and is found in the plasma of mothers and not in the milk that has been attributed to seropositive status in some infants who are anticipating treatment with Zolgensma. Unfortunately, because of the changes in rapid decline in SMA, we can't simply wait for those antibodies to decay over time, because it takes about 6 months for passively transferred maternal antibody to leave the infant. So this can inhibit access to treatment in infants with SMA. The IgM antibody, which is the one that's formed after the first exposure, does not cross the blood from the mother into the baby through the placenta, and also is not present in breast milk. So we have a paper that is about to be released that describes this issue and how we propose we might deal with it for infants who need early treatment, in the case of SMA, at least currently. Next slide. We know that antibodies are the main safety drivers of the main safety events we've seen. And in this paper published in January in the Journal of Clinical Investigation, we described 38 cases where we monitored antibody levels post-exposure, and in a subset of those patients prevented the antibodies through treatment with a drug that's used for treating other forms of B-cell disorders called rituximab. We were able to understand through this careful observation of the first few days and weeks after administration that AAV can activate one of the innate immune sensors called complement. Complement, it may be part of AAV's life cycle, but we know that this actually causes disruption of cell membranes, can injure cells as though they were pathogens. So the cells are bystanders, in effect, from the complement activation. That's particularly important in understanding the effects of complement on myocardial function, which has been responsible for most of the safety concerns in the AAV9 studies that were reported in this paper. But we do have a way to address this now. If you go on to the next slide, part of what we showed in the paper is that there's some by understanding the time course, and this is a bit complex, but focus at least first on the curve number 1, which is when AAV is dosed. In hours after the infusion, it reaches a very high level. There's a minor decline in the level of AAV in the blood over the first few days, but it persists in the blood for up to 2 weeks. And during that period, antibodies are formed, and you can see the first time we can detect antibodies of this early response type, IgM, is we're 0.5 at 4 days. That's what triggers the activation of complement predominantly. And by 6 days is when we see some of the biggest concerns about the complement activation. You can see curve 6, the orange line, shows the presence of D-dimers or fibrin split products. This is a blood product that occurs when coagulation happens within the blood vessels. So this is how we believe that the reactions also cause liver injury, myocardial injury, and because we can prevent the antibodies, we can potentially prevent all of the consequences of complement activation. So next slide. How do we go about preventing antibodies? They mentioned there are drugs used to combat B-cell diseases, whether it's rheumatoid arthritis or B-cell lymphoma. A very long track record of safety in a drug called rituximab, which is itself an antibody directed against the cells that ultimately make antibodies. And using rituximab pre-treatment, we found that we can block all of these innate immune responses, and at least for the period of protection from rituximab, which is about 3 months, we could even re-administer the AAV. So this is an important consideration. In lieu of single high dose administration of AAV, it may be preferable to split the dose and give it over 2 different sessions that are a few weeks apart. And so this is something we're considering for some of our patients with Duchenne who maybe have more susceptibility to that myocardial injury that comes from the innate activation of complement. We give this along with another drug called sirolimus, which helps inhibit the T cells that also cause anti-transgene effects and contribute to the liver injury associated with the AAV capsid or the shell of the protein. So those 2 drugs are given about 2 weeks prior to the gene therapy and continued, at least the sirolimus continued for 1 to 2 months following that. And we've been collaborating with several groups that are testing these therapies in adult patients, particularly trying to understand the effect on myocardial function for genetic diseases that cause heart failure. This will be a cornerstone of the study that Dr. Helderman is doing that I'll mention at the end of my 20 minutes. So next slide. When we block these antibodies, you can see that one of the blood measurements we do to assess the innate response is to look at the decline in platelets. The blood platelets are sensing OA's injury to the blood vessels, trying to prevent bleeding, and when there is an activation of the endothelium that occurs following AAV delivery, the platelets start to decline. And in the red line, when we prevent the antibodies from forming, you can see there's only a minimal change in the platelet count over the first 30 days. The bone marrow keeps up, and these platelets are usually restored, but it's an early indicator of what's happening within the lining of the blood vessels. So we think this treatment regimen at least is a first indication that we can enhance the safety of AAV vectors with this pre-treatment. The next slide shows what happens to that other marker, the D-dimer or the complement proteins known as SC5b9. When we pre-treat patients with this immune modulation strategy, we prevent accumulation or this clotting that occurs in the distal blood vessels. You can see the difference between the red line and the patients who didn't receive immune management. And that's true also of this membrane attack complex, SC5b9, which is part of the complement pathway. So next slide. So are there other ways we can achieve the same thing? And here's a list of the strategies that we've evaluated for reduction of anti-capsid antibodies. The drug I mentioned, sirolimus and rituximab. We've also studied daratumumab, or Darzalex, is a monoclonal antibody that binds to plasma cells and hopefully eliminates those cells. Bortezomib is a chemotherapy drug that's used to deplete antibodies in some cases of high antibody levels associated with B-cell tumors. And efgartigimod is a drug that's used to treat myasthenia gravis that we're repurposing to address anti-AAV antibodies. And I'll explain a little bit about that in a second. Next slide. When we consider the use of these for treating seropositive patients, it does take a while for the rituximab and rapamycin approach to let the plasma cells be depleted naturally, because we're not trying to treat those specifically. So that's step opportunity one. We have been working with a group here at the University of Florida, has been engineering versions of AAV with reduced immunogenicity. Although this may help be a workaround for pre-existing antibody, it doesn't change the response after exposure. This immunosuppression, number 3, targeting plasma cells, is Darzalex. And then the other 2 strategies are basically aimed at depleting antibodies, like I mentioned, the drug Vyvgart. So I'll explain how these might work in the case of treatment for Sanfilippo at the end. If you go on to the next slide. What are the population prevalence of antibodies? In one study done by Sarepta of over 100 boys with Duchenne, they found about 17% had disqualifying antibody levels. What's important about this is that they used a much higher threshold than in other studies, particularly ones where AAV was being delivered into the spinal fluid, or that commonly used for SMA. So when you increase the threshold for exclusion, fewer patients are excluded. This is a little more lenient, but even these higher antibody levels were not associated with adverse events. So we think that maybe other products could follow this same guidance. When we look in the real world, though, even at this threshold of 1 to 400 antibody titer, about a third of patients of school age are excluded from receiving the commercial product. Next slide. And I show you how those persist. This varies around the world. In the companion to the plain language summary paper, we looked in a lot of detail at the various assay methods and what it means to be antibody positive. You can see that there are regional differences that we think maybe reflect the immune response profiles of the people as opposed to the actual exposures that might occur in those different places. Next slide. How do AAV antibodies impact therapy? For one, we think probably the principal way is they block the binding to the cell surface, and the AAV cannot then get inside the cell where it delivers the payload or the gene to the nucleus. There are some antibodies that actually facilitate this reaction, but most of the ones that we've identified related to AAV actually block the AAV receptor. The other possibility is that many antibodies aggregate with AAV and traffic it away from the target tissues, which might be the liver, the CNS, or the musculature, and that is a major component of this mechanism as well. Next slide. This is a lot more detail about these various drug approaches. I've explained some of them already, and some we've not yet tested, but the principal approach we're using is this rituximab method, which I described. So that's the first panel in that figure. So next slide. Now, the other opportunity is to prevent the recycling of antibodies and thereby lower all antibodies. This is the principle of a drug called efgartigimod, which is a part of an antibody. It's the FC portion of an IgG. When that is bound into the pathway for recycling of AAV, it lowers the recycling of all antibodies in the circulation, and this is found to be effective in treatment of a form of autoimmunity called myasthenia gravis. Next slide. So what we intend to do is use Vyvgart in the treatment of some boys with Duchenne who have pre-existing antibodies that would prevent them from receiving the commercial product, as well as in a cohort of patients who've already been treated with gene therapy. This study design is shown on the next slide. If you go ahead, we're basically, one forward, yeah. When we use efgartigimod in the preclinical model, you can see that 4 doses of efgartigimod given -14 days, -7 days, and then in the middle of the administration of the vector at day 0, we can prevent the antibody formation for up to 70 days. This may be a really important way to prevent adverse events, and it actually enables us to give a second AAV dose, which we've done here at this 70-day time point. So we're about to start this study in boys with Duchenne. If you go on to the next slide, this is work supported by 3 foundations, the MDA, Parent Project Muscular Dystrophy, and Cure Duchenne. It's based on this preclinical data with a microdystrophin where we showed that anti-CD20 or even in combination with Vyvgart is the most effective at preventing the immune response compared to no immune suppression. This also has an effect of increasing the effectiveness of the treatment vector when there's pre-existing immunity. And we know now that actually we can double or even triple the expression levels from the vector by preventing the removal of the vector by antibodies. Next slide. This is a brief overview of the 2 different groups that will either have elevated titers from natural exposure or post-gene therapy, 6 boys in each group that will receive 3 different doses, at 3 different dose levels of antibodies to see whether at what threshold we can lower the antibodies. All of these passively immunized or natural exposure patients have either low, medium, or high antibody levels. And so the second group will consider the ones that have very high, greater than 100,000 units or up to 1 million units of pre-existing antibody post-exposure. Next slide. The results of this study will be available fairly quickly. We hope to enroll all these boys over the course of 2 months, starting early in January. And it really requires that they each receive 4 infusions of efgartigimod, and then we can test immediately after the effect on the antibody level. So the intervention ends in just 4 weeks, and we'll know how well the antibodies were lowered, similar to the studies that were done for myasthenia itself. Next slide. And I'll just very quickly about this part. We can also consider ways to specifically lower antibodies with a antibody-specific degrader molecule that's being developed by Biohaven. So this gives you a summary of what this looks like. We've identified target binding portions of the AAV vector. It will be linked to this molecule that drags the antibody into a degradation pathway in the liver. And so this is work being done in collaboration with Biohaven. Next slide gives you an idea of where they are with this. Right now, they've developed the platform, very fast at developing these molecules, and then while they're doing that in some pan-IgG syndromes, we'll be developing this second-generation antibody-specific degradation. Next slide. This just shows you, again, a summary of what is involved to get this degradation to happen. So these are called modes, or a terminology used by Biohaven for this modality. Next slide. So I want to take the last minute and talk about a program that we've begun with Dr. Coy Halderman here at the University of Florida for many, many years. From the time he was working with Mark Sands at WashU, he's been studying MPS IIIB. And if you go on to the next slide, Coy's approach is with an AAV8 vector that has some specific mutations that enhance its delivery in the brain or when delivered into the spinal fluid. And the assessments are all shown here. Some important outcomes related to survival and behavioral phenotypes. Particularly important is the auditory evoked brainstem response, or the ABR, which is abnormal in Sanfilippo. And that's one of the key readouts that can be done in mice. So if you advance to the next slide, I'll just say one bit of overview of the findings so far. You can see that the 3B mice that don't receive treatment don't survive as long as wild-type mice, shown in the black line, and that the treated mice in either route of delivery do very well and almost survive really essentially to the level of the untreated wild-type mice, and that they have improvements in the auditory brainstem evoked response that are equivalent again to the wild-type control mice. So very encouraging early results. And the grant we received, if you go to the next slide, you can skip over this one. This just shows that actually the levels of, it was mentioned, if you go back, that when we look at NAGLU activity, when NAGLU activity is restored, you can see that compared to controls, all the treated mice have NAGLU activity above the threshold of unaffected controls, and that when NAGLU activity is normal or above normal, the biomarkers in CSF are normalized of HSPG. So next slide just gives you a quick summary of what we're doing in this grant funded by the NINDS. This is part of the Nanotherapeutics Network or Blueprint grants, and these will enable us to complete non-clinical studies to support an investigational new drug application in Sanfilippo, and the milestones are listed here. And this grant been underway for the past few months, and we meet every week and really are making great progress towards initiating this IND for Sanfilippo B. And there, and I suppose we'll take questions at the end. I do want to thank all the funding mechanisms that have supported this work, as well as our team at the University of Florida.
Okay, good morning, everyone. My name, first of all, thanks to Glenn for your really incredible introduction. I was really touched by it. And for Adam and for Dr. Byrne, both for just also laying out the framework for today, and for Barry for just such an incredible and inspiring presentation. You've actually helped me shorten mine, which I think is really great. So just so everyone knows, my name is Rafael Escandon. I am a bioethicist, but also a clinical scientist. And actually, Adam and I have been colleagues for the past 10 years in 2 different companies. I am a consultant now, but I do actually consult, just as a disclosure, for BridgeBio Gene Therapy, Adam's employer. I wanted to talk about the bioethics or some broad bioethical principles and apply them to what's been done in gene therapy for monogenic diseases over the past decade or so, maybe a little bit longer, and talk about things a little differently than has been done so far. The inspiration for this presentation actually was a conversation with Cara several years ago, and we were talking about really the application of gene therapies and participation in gene therapy clinical trials early on. And her remarks have always stuck with me and have now taken several iterations of several presentations at annual meetings. Another comment I got when I presented a similar presentation to this at the American Society of Bioethics and Humanities last year, when I discussed what I thought was the problem, it was interesting. Someone said to me, "Well, that's just research." I don't really think so. I think we can think about things a little bit differently. And then throughout the regulatory documents and the regulations of the FDA and the EMA, and really about biomedical research in general, as it's applied to pediatrics, there is a value of what we call generalizable knowledge. Anytime we do research in humans, and in particular in patients who are considered vulnerable, like children, there is always a balance between obviously risk and benefit. You've heard about that, but also the prospect of directly benefiting from participation in research, and weighing also this other concept of generalizable knowledge, generation of generalizable knowledge that can be applied to the entire disease state, for example, or to potentially all patients down the road, that you get some answers to some questions that you didn't that didn't exist before, but that the patient themselves, the participant themselves, doesn't benefit directly. So that's this concept of generalizable knowledge, and this population of patients now that we've accrued over a couple of decades of doing gene therapy. There is a population of patients who haven't directly benefited, but who have contributed enormously to the advancement of gene therapies as a potential modality. Next slide. So I just did a word cloud here. These are some terms to bioethicists and clinical ethicists and clinical trialists that are very meaningful. But in general, when you do research, it's always a balance between the prospect of benefit, and that would be efficacy for a gene therapy, and risk. And risk would be, of course, the possibility that something adverse would happen, whether it's temporary or permanent. That's the real balance. But getting down into the nitty gritty, there are a lot of other issues, a lot of other elements that come into play for gene therapies versus a typical small molecule drug research trial. And so those are, as you've heard Adam mention and heard Dr. Byrne mention, there are long-term implications from participating in a gene therapy trial that don't exist for a drug. And we'll talk about immunity is one of those things. The inability to redose is similar, but also a major thing. Following up patients for 5 years to 10 to 15 years is another difference, and that creates a significant research burden. So I'm going to go into a few of the differences I think between that we've learned over the past 2 decades that differentiates gene therapy research participation from a typical drug trial. Next slide. So I include this slide because back when I was an undergraduate in the late 1980s, early 1990s, so 30 years ago now, the quote up at the top by Dr. Francis Collins saying, gene therapy has the potential to revolutionize the way we treat many genetic and acquired diseases. One of my professors as an undergraduate said, and I'm from the East Coast originally, he said, there's this new thing called biotechnology that I think a lot of you are going to ultimately be involved with one way or another. There were a lot of pre-med students, but a lot of scientific students too. And so I just laugh about how sage that comment was back in the day. But I think one of the things about gene therapy that is so interesting and actually a little bit different than I expected, and this diagram is about 25 years old now. But this concept that gene therapy, if you go straight down to the bottom, the potential of a therapeutic product is actually only one area of the application of gene therapy science. There is, if you go to the left-hand side of the slide, there's a diagnostic, there's a preventive, there are population analyses that can be done with genetic information. Similarly, if you go to the right side of the diagram, you can understand by by elucidating various protein pathways and signaling pathways, you can understand the basis of the biologic defect better and actually develop a small molecule drug therapy or a biologic therapy. So it's not just, the Human Genome Project and gene therapy aren't just focused on developing a specific one-time gene therapy. There are also many other applications that are extremely important, and I think some of that knowledge is what Barry just described in his really, really amazing comprehensive presentation. So next slide, please. So I just did a quick search on a repository, a data repository that a lot of folks are familiar with, and I just tried to really put a funnel together to show what the gene therapy research landscape looks like. And this is from about 6 months ago, but you can see that there are literally 2,000 or so. That number doesn't change too much. Gene therapy trials that are true interventional trials. Most of those are early stage trials, and about a quarter of them are active. But I think in the end of the day, what you can see is that as that funnel tapers down to the bottom, there are a large number of them that are industry sponsored that often get, because there's funding or there has previously been funding by the industry to sort of hasten development, hopefully, of some of these gene therapies, that things go quickly, but that's really a small proportion of the number of actual active gene therapy trials that are happening in any given time. Next slide. So I won't go into this. This has been presented by both Adam and Barry. I think, in the end of the day, this is just a simplified version of how an AAV, an in vivo AAV gene therapy, is introduced into the body and and travels to the target cell and the cell nucleus, and then ultimately expresses the protein that's either defective or isn't expressed at all in the target cell. That's the basic concept here for AAV gene therapy. Next slide. So now I want to transition a little bit to what I view as the differences between drug and gene therapy research, and in my opinion, how similar that gene therapy itself is actually more similar to pediatric organ transplant than it is actually to the typical to rubric of drug and biologics trials that really all of clinical research sits upon. So I won't go into every little detail of this slide, but just to say that some of the big differences are that at the moment, these are single and permanent interventions. That gene therapies are that very much like an organ transplant. Once you have gotten it, you're potentially permanently ineligible, or it's very difficult to be eligible for a second treatment. That's a little different for organ transplant than gene therapy, and based on Barry's presentation, that may be moving in the right direction for pediatric gene therapy, which is fantastic. Obviously, you have at the moment immunomodulation required for gene therapies as you do for organ transplant. There are years to decades of follow-up that aren't required of drug trial participation, and I think one of the things from a bioethicist perspective is that when you're talking about pediatric research and you have given this one-time intervention, you have to, and you're participating in a trial for say 5, 10, 15 years, much like an organ transplant. Your agency, your capacity changes over those times, and so from the consent process and from the development perspective, you have a little more, maybe a much more complex set of decisions to make. So in general, though, all this comes down to the fact that currently, at least with the historically, we've been dealing with a set of modalities in gene therapies that can't be turned up, can't be turned down, can't be turned off, and can't be redosed. And again, understanding that Barry's presentation maybe gives us a much brighter future for maybe all of those elements. But at the moment, we're in a place where all of those features unfortunately apply, and so that's very, very different than participating in a small molecule trial, and that's the foundation, and that's the rubric that clinical research is built upon. So I just want to encourage parents who are considering participation in gene therapy trials to really sort of consider that very carefully and think through all the implications as best you can while balancing the prospective benefit and the possible generation of generalizable knowledge if unfortunately your child doesn't benefit directly. Next slide. So I've really covered this, but these are just the types of the different types of research that the FDA categorizes different types of pediatric research into. They really go in sequence from the probability of benefit and low to no risk to balancing that, as I mentioned, the probability of direct benefit against the possibility of only having generalizable knowledge that's generated for the whole population. So there are 4 different flavors of how research protocols are reviewed by institutional review boards, and we categorize research into one of 4 categories there. Next slide. So this is another point that I want to make here is that in my industry career, I did spend some time working in early stage oncology development, and so that's more or less the same design that we use for the early development of potential gene therapies. So say a first in human trial, this is a pretty typical design. This is not every trial is like this, but it's a pretty typical design where you start at the bottom left of the slide here, start at a relatively low dose, maybe you dose 1 patient, you assess them for a period of time, get them through an acute phase, and then if things seem to be going in the right direction, that maybe you're observing some potential benefit through a biomarker or through something else, you may dose, say 2, 3, 4, 5 more patients potentially at that dose level. But the idea here is to get answers quickly. Hopefully you're right on the first dose because of all the reasons that I mentioned, but sometimes you're not. It's a really, Adam can tell you, he and I have been through this together, and anyone developing a gene therapy, choosing that first dose, translating the data from your animal models into a human protocol, and picking that first dose that gets plugged into a model like this is one of the most difficult and agonizing decisions that you can make, because at least from today's perspective, that patients that's their only opportunity to be dosed. So you want to get to an effective dose and without compromising safety as quickly as you possibly can, and you want to dose as few patients at ineffective doses as you can as well. But without any clinical experience, it's very difficult to hit the bullseye. So this is in general how gene therapy, early stage gene therapy programs are developed. Next slide. So I'm just going to apply that model to this, and this is Zolgensma, which you've heard a bit about. And so think about the left-hand panel of this slide as that first dose level, the bottom left of the slide. And so you can see in the red circle, the vertical axis here is the functional score, and that's more or less the marker to show that the patients are benefiting. And you can see at this low dose, at the cohort 1 dose, that the 3 patients who were dosed really didn't improve at all. And so they have generated generalizable knowledge to say that that dose is too low, at least based on on the vertical axis CHOP INTEND score. So they raised the dose. And remember, go back to the slide that I showed you for the design. Now dose cohort 2, transformative, incredible. Every patient dosed except for one showed a remarkable response here on that CHOP INTEND score. So this is an enormous success. Another piece of generalizable knowledge that was important generalizable knowledge that was generated by the one patient that you see in the pink circle there, however, is that there seemed to be a very time-related dosing window outside of which patients don't benefit. Now, that's very familiar to the Sanfilippo community. It's very familiar to any community that deals with cumulative neurodegenerative programs, and so that's included in Barry's presentation as well. It's a very, very important element that we need to think about, but sometimes is difficult to calculate when you're in the early stages, when you only have animal data to consider. So these are patients to whom I believe we owe a great deal of gratitude and debt, because they have provided just incredibly important information for the further development of these programs. And my personal belief is that we undervalue these contributions. Next slide. So this is just a little analysis I did for the ASGCT last year in 2024, just to try and calculate patients who in red, on the red far right column in red, are the number of patients who received doses that were below the ultimate dose that was found to be effective. So these are the total patients who were who provided what I would call generalizable knowledge only and didn't directly benefit. You can see that in some cases, companies got lucky, which was fantastic. That may be related to the modality. An ex vivo lentiviral intervention, for example, may be a little more amenable to, for many other reasons, toward picking the right dose the first time. But the bottom line is hopefully we're getting better at this. Hopefully we're limiting the number of patients as best we can who get ineffective doses in early development for gene therapies. Next slide. So again, as I pointed out earlier, it is especially compelling in conditions where they're rapidly progressive and disabling, permanently disabling, and rare, that the parents' risk tolerance for investigational gene therapies is really factored into the equation. And what I mean by that is look, if there's no other option, and over the decades, I've talked to many parents and many family members who feel a lot of pressure, and I think Barry mentioned this a bit too, is that you feel a lot of pressure to participate because doing nothing is much worse than looking at an opportunity and saying, well, I have misgivings about that. That might not be right for my family. But if it's the only thing going, I have to do everything. And I'm so sympathetic to that. But I also feel like it's really important to understand fully as part of the consent process, but also for the child, but also for the whole family and for caregivers to really appreciate the fact that this is a really long-term commitment, and it's one that will have many implications over time, but that it is possible that that a benefit won't happen, and it's a very different situation to be able to get redosed or to even be able to participate in a gene therapy trial or even a small molecule trial down the line. I'll talk about that in a moment. But in the end of the day, I feel like companies can also invest in ways of thinking that are slightly different from the typical rubric, which is are there things that we can do as companies, or that companies can do, to begin that early development process with doses that are more likely to provide a direct benefit? I think there's a real drive to be able to, and a real ethical rationale to be able to do that. Similarly, fewer patients per dose escalation cohort. Sometimes it's really hard to tease out whether a safety finding is spurious or whether it's characteristic of, related to that dose given. And so that's a really challenging aspect too. But in the end of the day, I think that if we can find a way to have fewer patients per dose escalation cohort that are below the activity threshold, the efficacy threshold, the better will be. And then one of the ways to do that might be, again, it's important to balance safety, but can we do larger dose escalation increments? In the end of the day, though, I think that parents are the only ones who can answer the question about what their risk tolerance is for their family. And I don't think that sponsors can do that. I don't think that that regulators can do that. I think that is a conversation that clinical investigators and families need to have, perhaps in a bit more pointed manner than happen in typical drug trials, because these are very different for the reasons that you've heard me say. Next slide. So finally, really, I think, again, my effort here is to also bring attention to those participants who have provided this generalizable knowledge that is so important, that continues to move individual programs, but the entire cadre of gene therapy developments forward. And I think that that to date, those patients have been underappreciated, whereas the contribution they've provided has been enormous. I might even argue as enormous as those who've benefited. So I think that the idea that equal and fair recognition for their continued long-term participation by everyone involved, investigators, site personnel, sponsors, I think we can't say thank you enough to those families and to those patients. I think there should be special recognition, and this is not occurring, and I think it really should in study publications of clinical trials. I think that as well, early completion or reduced assessment burden for patients that clearly haven't benefited, to the extent that safety isn't compromised, is a really important thing to offer them. I think fewer limitations on protocol exclusions for prior receipt of gene therapy in, say, small molecule or biologics trials. This is very related to sponsors and investigators. I think greater consideration and greater openness needs to be thought about for that. I think Barry mentioned that a little bit in his presentation, and I'm fully with him. And then I think rapid publication and full data sharing with stakeholders, regardless of the competitive landscape or commercial plans in the entire enterprise of gene therapy, needs to happen better. That will cause all boats to rise, and I think expedite this. So that requires a lot of cooperation and a little different way of thinking than companies and academics and government agencies are accustomed to. So next slide. So back again to our word cloud here, and really, I think what I want to just impress on folks again is this balance between beneficence and generalizable knowledge, research burden, risk tolerance, and the future are really important practical and ethical considerations that we need to think more carefully about as we get to these next generations of gene therapies. So just want to say thanks and appreciate the ability to participate in this whole session. So big thanks to Glenn and Cara.
Okay. Good morning. Good morning, everyone. Okay, so first of all, I would like to thank Glenn, Cara, and the organizer for inviting me. I recognize, as I think all of us, your incredible and continuous support to advance research in Sanfilippo. Okay, so this is the title of my talk. So I will talk about a new combination therapy for Sanfilippo disease. Next slide, please. So there is an unmet medical need, as all of us know. Currently, in fact, there are no therapeutic options that can treat the neurological manifestation in patients with Sanfilippo. Next slide. Possible therapy for Sanfilippo and other neuropathic MPS, mucopolysaccharidosis, are based on the correction of the primary inherited defect by gene therapy or enzyme replacement therapy. Next slide. And there are some of this therapy are in clinical care. Enzyme replacement therapy are available for some form of mucopolysaccharidosis, and also hematopoietic stem cell therapy is available. However, none of them correct neurocognitive decline. Next slide. There are different clinical trials that are ongoing for different forms of neuropathic MPS, including Sanfilippo. Some of these clinical trials target the CNS. They are based on enzyme replacement therapy with the blood-brain barrier penetrant enzyme, or based on AAV gene therapy, AAV9 gene therapy. Next slide. Of course, these therapies are promising. The main advantage is that they can treat potentially the primary cause of disease. But there are several challenges and also disadvantage of this therapy. They must be developed for each single enzymatic defect, of course, and then several clinical trials based on AAV gene therapy or on enzyme replacement therapy failed because of the difficulty in achieving and maintaining the therapeutic threshold level of the corrective enzyme in the target tissue, and particularly into the brain. And then the high dosage and the repeated administration of the therapeutic enzyme or the viral vector are not only costly, but can be potentially toxic and immunogenic, as has been discussed also by previous presentation. Next slide. So an alternative approach to treat the MPS is based on targeting pathways that are downstream the primary defect. So basically this alternative approach can inhibit secondary pathogenic mechanism. Next slide. The aggregation and deposition of amyloid protein, such as alpha-synuclein, huntingtin, beta protein, or tau protein, has been shown by our group and also by other laboratories in post-mortem human brain sample from Sanfilippo and other MPS patients, and has been observed also in the brain of several mouse models of mucopolysaccharidosis, including Sanfilippo. And in some studies that we published some years ago, we were able to demonstrate that alpha-synuclein is the main component of this amyloid deposit. As you can see here, the immunohistochemistry with antibodies against alpha-synuclein strongly evidence the aggregation of this protein inside the soma of neurons. And the alpha-synuclein staining colocalized with the thioflavin positive aggregates. Thioflavin is a dye that stain different amyloid protein, including alpha-synuclein. Next slide. And basically, we were able to demonstrate that this aggregation and the deposition of amyloid protein contribute to the neuropathology in several forms of neuropathic MPS, including Sanfilippo. So we started to use a drug belonging to a group of molecules known as molecular tweezer. These are potent broad spectrum inhibitors of protein aggregation. CLEAR1 is the lead compound of molecular tweezer. These are basically small molecules that can inhibit the self-assembly of several amyloidogenic proteins, including alpha-synuclein and beta protein. They act by a unique process-specific mechanism by binding a specific lysine in amyloid protein and inhibiting the formation of beta sheet and therefore of amyloid aggregates. Then they rapidly concentrate into the lysosome. Does this drug have effects in vitro and in vivo on different models of neurodegeneration? So basically, these drugs have been developed by other group that demonstrated that this drug works and can protect against neurodegeneration in different models of neurodegenerative disease, such as Parkinson and Alzheimer. And specifically, these studies has been done in the lab of Gal Bitan, that is our collaborator, and Thomas Schroeder in Germany, that who is the chemist that developed these drugs. They are potentially safe, so side effects can be observed only at very high concentration. I would like to underline that this drug have been used only in preclinical studies, so never used in clinic. Next slide. So when we used this drug in an MPS IIIA mouse model, we were able to demonstrate that the aggregation of amyloid that you can see very well here in MPS IIIA brain by thioflavin staining in the upper panel can be reduced by administering the drugs to the mice. And this can be also observed by using a specific antibody against the specific component of amyloid, such as alpha-synuclein and tau protein. And the accumulation and the aggregation of this protein was strongly reduced upon the administration of CLEAR1 to MPS IIIA mice. So the CLEAR1 drug has been administered by using intraperitoneal injection. This is a continuous injection, of course. It's a daily injection. The drug is able to cross the blood-brain barrier, so this is why we see the effect into the brain. Next slide. And the reduction, and this is very important, of course, the reduction of the amyloid aggregate was associated to the amelioration of different neurological signs, such as neuroinflammation, and also the behavior of mice. Next slide. Next. Autophagy blocked, so the stress of the autophagy lysosomal pathway is a key event in many neuronopathic mucopolysaccharidosis, including Sanfilippo, and what we were able to demonstrate is that the neuroprotective effect of a CLEAR1 was mediated by a reactivation of autophagy flux. We used the LC3 marker in the upper panel. This is an immunofluorescence against LC3. As you can see, there is a lot of staining in MPS IIIA brain that was strongly reduced by using the drug. And there was also a reduction of the lysosomal compartment. Next slide. So this means that the deposition of amyloid protein can affect in some way the autophagy lysosomal pathway, and this trigger also a vicious cycle that contribute to the CNS deficit. And by using the CLEAR1, we were able to inhibit amyloid deposition and therefore the triggering of the vicious cycle. Next slide. So driven by this data, we challenged the CLEAR1 also in other neuronopathic MPS, and specifically in MPS 1 and MPS 3C in collaboration with Alexi in Canada that provided us with the MPS 3 mice. So we treated the mice with the drug at the same dose and same frequency that we used in MPS 3 mice, and we were able to demonstrate that also in these mice the drug was able to reduce the neurodegenerative signs of the disease, so the neuroinflammation, lysosomal enlargement, and also the behavioral abnormality. And this data has been published recently in molecular therapy. Next slide. Using the molecular tweezer to target amyloid protein and autophagy dysfunction can be considered as a novel therapeutic strategy for neuropathic MPS. So the challenge, of course, is that this molecule has been never tested in human, but only in the preclinical model. There are several advantages that can be applied to multiple MPS. We already demonstrated the proof of efficacy in model of MPS 1, 3A, 3B, and 3C. The molecular tweezer target specific pathogenic mechanism involved in autophagy stress and neurodegeneration, and rapidly concentrate into the lysosome. And this make this drug particularly efficacious in the neuropathic MPS and allow, in principle, them to achieve therapeutic effect at low doses. Next slide. Then we speculate, because molecular tweezer can target a secondary toxic storage, so the storage of amyloid. So we speculate that combining molecular tweezer with the gene correction, with gene therapy, can in some way add a beneficial effect on the therapy. And this is something that we studied in MPS 3A mice. Next slide. So we combined a gene therapy mediated by AAV with CLEAR1 in mice. So gene therapy was applied at 2 months of age, and CLEAR1 was applied continuously from 4 months of age. And here you can see that the combined therapy reduced neuroinflammation and the lysosomal enlargement, that are 2 key markers of neurodegeneration, more efficiently than monotherapy. Next slide. We were also able to demonstrate by evaluating the behavior in mice that combined therapy correct behavioral abnormality more efficiently than monotherapy. Next slide. And so the conclusion is that the molecular tweezer, the combination of molecular tweezer with the gene therapy result in a synergistic therapeutic effect. Next slide. Next. And therefore, so targeting amyloid protein deposition and autophagy stress by using molecular tweezer can achieve therapeutic synergy with the AAV gene therapy and potentially also with the other replacement approach. And of course, this have a potential positive impact on the clinical application. Next slide. So finally, I would like to thank all the people that were working on this project, and of course the sponsor, including the Cure Sanfilippo Foundation and the Sanfilippo Children Foundation, and Sanfilippo Fighters that supported this research project, and Fondazione Teleto and Orphan Disease Center. So thank you very much to all of you.
Hello, I'm Cara O'Neill, Chief Science Officer at Cure Sanfilippo Foundation. I conducted this study with Elise Drake, Dr. Elise Drake, who is an incredible parent volunteer and professional that helped with the data analysis. As you've heard already this morning, there is some amazing scientific progress happening in gene therapy and of course, other areas of research as well, which you'll hear more about in the conference. But as we inch closer to the possibility of gene therapy approval, it's important to check in with our community about what they need to make informed decisions about future therapeutic options. Next slide. Knowing that the FDA was planning a listening session to gather perspectives on gene therapy, we decided to go out directly to the Sanfilippo community in order to get the most current information about how they were thinking about gene therapy and what factors are important to them when considering treatments for their children. To do this, we put together an anonymous survey and asked parents to respond to 7 free text format questions. These questions were adapted from the topics that were posted by the FDA and available as part of their pre-meeting materials. We distributed a link to the survey via email and through online Sanfilippo-specific social media forums. Next slide. We had a very nice response, and 44 parents filled out the survey, reporting there were 2 sibling pairs, so reporting on 46 children, and most of the children were in the preschool age range, 3 to 5, followed by the 9 to 10-year-old range, but we did have responses across many of the age groups and inclusive of adult children. Next slide. As we might expect based on prevalence, most of the children had Sanfilippo type A, followed by B and C. We did not have respondents with Sanfilippo D. And most of the respondents were living in the US, although about 27% were from outside the US from a variety of different countries. Next slide. So the first question posed by the FDA asked about what risks or uncertainties people would consider when deciding to receive an approved gene therapy. But we also wanted to incorporate another scenario, and that was the scenario of an experimental gene therapy from a clinical trial, because that's more relevant to our current situation in Sanfilippo. And also we wanted to understand if there were differences in the decision-making and what information parents needed. Now, of note, of the parents that responded, about 40% of them, their children had been in some sort of a clinical trial. And 1/3 of those actually had received gene therapy. So really unique circumstance to hear from those who actually had experience with gene therapy. But for the majority of folks with Sanfilippo, this is going to be a hypothetical experience where we're asking them to really think about what if. Next slide. So naturally, parents want to be armed with information about risks and benefits when considering any healthcare decision for their child. And we see that's the case, whether the treatment is under an experimental clinical trial scenario or an approved gene therapy scenario. However, we saw that come up much more under the experimental scenario, and that just is, makes sense, because I think, parents recognize that if a drug is not yet approved, that there is naturally going to be more uncertainty and risk involved with that. Next slide. We also saw a marked difference around the consideration of burden between approved and experimental therapy scenarios. The aspects of burden that came through in parent responses were things like they wanted to know about the invasiveness of a treatment or other procedures associated with the long-term follow-up of those treatments, the frequency of travel and how much time they would need to be away from home or staying in a hospital. They wanted to know about the other kind of unaccounted for costs of participation, lost time at work, and impact on other family members. So clearly the risks and burden of participation are more of a consideration when you're in an experimental clinical trial scenario. Next slide. But despite the risks, burdens, uncertainties, and so many other categories of responses, about 30% in an approved scenario and 27% in a clinical trial scenario said that they would still accept any risk in order for their child to receive a gene therapy. And we heard that in Dr. Escandon's talk, that this is common in devastating diseases with no therapeutic options, but still important to realize that, yes, indeed, this is how families with Sanfilippo perceive their decision-making. Next slide. Question 3 focused less on risk and uncertainties and simply asked parents what they needed to know about gene therapy. So a bit more open-ended, and the responses were really broadly distributed across the categories. To sum it up, we want to know everything that we can know. Next slide. But I want to focus on the other factors kind of category and what that meant to parents. The things that they wrote were things like, we want to know how well does the treatment reduce heparan sulfate levels? We want to know, what did they find in the preclinical animal studies? And what were the characteristics of the children or people who were included in the clinical trials? And how does that relate to my child? Next slide. Question 4, I thought was a really interesting question because it asked people to think about if there was more than one treatment option, what information would they need to decide between them? And we saw a significant number of responses that fell into different categories, effectiveness and, what are those outcomes, side effects, and so on. Next slide. But weighing information about potential benefits and burden of participation were the most noted factors that they needed. And families really wanted to understand, what could this treatment do? And when they were thinking about benefit, things that they would say were, what did this do to stabilize disease process? What did this do to slow disease process? How did this impact different aspects of quality of life for my child? And very much so, what would it take for my child to receive this? What is our family going to have to go through? How much of a burden on our daily life? Next slide. Question 5 was focused more on this long-term follow-up scenario and what would prompt parents to participate or not participate in that. And we heard about the context and the need for long-term follow-up in gene therapy studies in particular. And for AAV-based products, that tends to be around 5 years. For lentiviral, longer follow-up is usually needed. And these studies are often called post-marketing registries. So you'll hear the term long-term follow-up, but in an approval scenario, it's called post-marketing registries. These are particularly important when drugs are approved under a conditional basis or an accelerated approval pathway. They're important for understanding safety, but in Sanfilippo, they're particularly important because in a short-term study over 2 years or even 5 years, it can be hard to understand the full spectrum of benefits. Those are things like, did the treatment preserve walking ability longer? Did it preserve the ability for the child to eat and drink by mouth longer, an impact on other aspects of daily life that take a long time to bear out and really see a difference. And these are things that are happening outside of cognitive testing. So really important things for families that they would want to know about a treatment. So what factors impact parents' willingness to keep following up? Next slide. Back one slide. Again, we want to know what the benefit of continuing to participate is and what is the burden on us. Okay, next slide. Parents are thinking about all the things that you might expect: privacy, data sharing for research. But part of this benefit in participating in these studies is wanting transparency and sharing back of findings with the participants themselves. And, despite all of that, they're recognizing that, they may not be receiving a significant benefit, but they are really wanting to help children in the future. And, we heard Rafa talk about that earlier, but this is, truly something parents are saying. Next slide. The next question is more around operational aspects, and I won't go into detail here, but you see on the slide the similar themes were coming out. Some parents did offer suggestions on ways to reduce burden of participation in long-term follow-up, and those are things like limiting the travel time and distance and frequency, increasing ways to participate remotely, and having easy ways for them to input data or fill out surveys. Next slide. And lastly, the FDA wanted to hear information about long-term effects, what parents wanted to know about that, and we saw varied responses across many categories. But next slide, by and large, benefit considerations were top of mind and what they mentioned most. In the end, what can we expect to be more positive or stabilizing about this treatment versus what we know will happen in Sanfilippo? Next slide. And then I just want to point out, though it's a lower percentage, it is something that we saw respondents answer across questions or mention across the questions, and that was availability of access to other therapies and how receiving a gene therapy might impact their ability to be in other clinical trials, receive other treatments in the future, and how those treatments might interact with one another. And so these are really important details to share with families as they're making their decisions. Next slide. So just high-level overview slide here, which you can read. But I just wanted to point out that parents really do want to know the details. They don't want to be given a gloss over of here's the efficacy, XYZ. Did it work? Did it not work? There's a lot of loaded bias in did it work, because how something works is very specific to the child and the priorities of the family. And so families really want the information so that they can make their own risk-benefit calculations that align with their individual circumstances. Thank you so much. And thanks to all the other speakers for the great information. I think we're going to go to Q&A now.
Great. Well, thanks, Cara. Thanks, Dr. Fraldi, Dr. Escandon, Dr. Byrne. And we do have a few questions here. And I know we don't have a ton of time, but there's a question here for Dr. Byrne. And the question says, about 40% of the population carries a variant of factor H. Have you gone back to your study patients to see if there is a variation in the antibody response in those patients who carry the Y405H genetic variant in factor H? And the follow-up question to that from this person is, would drugs that target complement might be beneficial? So do you want to take a crack at that?
Yeah, can you hear me all right? We have begun doing whole exome sequencing of all candidates for gene therapy to identify risk factors. We don't have enough, I think, examples to do a proper GWAS analysis against that exomes or whole genome sequencing. Some studies do include sequencing of all the relevant complement genes to see if there are any mutations. And I'm aware of one example where there was a mutation that affected TLR9 signaling that was strongly potentiated the antibody response. So it's a great point. There may be concomitant risk factors, and I think we're really trying to learn what those might be to manage those risks, prior to exposure to gene therapy.
Got it. Thank you. And in terms of, I think the follow-up question on drugs that inhibit complement activity, I know people have talked about drugs like eculizumab, which are used in other settings that can, in other diseases that are characterized by high complement activation. To your knowledge and in your experience, have people used this to try to either treat or prevent complement-related reactions to gene therapy?
In one of the Duchenne studies, where we enrolled the majority of the patients, after finding TMA events in the majority of the participants, we implemented a prophylactic eculizumab treatment. And since that's very downstream of the activation point, which is at C3, the kind of pivotal point between the classical and the alternative pathways, it didn't have a dramatic effect at lowering the cleavage of C3 in particular. And that's where some of the risk factors come from, because C3A and C5A are two molecules that affect tight junctions and allow water to come into tissue. So it causes generalized edema because the vector does go to all tissue beds. We think that the C3 inhibitors would be more effective. And there are also some small molecules inhibitors of just the alternative pathway. Those could be used in combination with the antibody blockade strategy. But those C3 inhibitors were previously investigational. And so that added to some complexity about using them in the context of a clinical trial. But now Empaveli is approved for PNH, an adult disorder. So there's not a lot of pediatric data, but we think it would be potentially a useful adjunctive therapy.
Thank you. I had a question for Dr. O'Neill, for Cara. A very, very interesting presentation, and really to understand what families are worried about and the risk tolerance. And one of the really interesting points that you made is people really want to have their data. They want better data sharing. And you were part of a really amazing presentation earlier this year at the World Meeting around data sharing, trying to get different sponsors to share data. What do you think is the biggest obstacle for giving patients back their study data from clinical trials?
I think there is a perception that it will somehow influence their responses to clinical outcome assessments, or observer reported assessments. I think that's the worry. I think the use of biomarkers and other objective measures, as we start to see those utilized more, will solve some of those concerns, and really is something that we need to be putting a lot of effort into, is exploring additional objective outcome measures.
Thanks, Cara. And I guess, for Raf, you've spent a lot of time, in your past and even part of your current life is the operational side of clinical trials, conducting clinical trials. And now you spent a lot of time thinking about the ethics of clinical trials, and especially for gene therapy. What is your perspective on what Cara just said? How about sharing data? How might we operationalize this in a way that might be more palatable to sponsors, but also give the community back what they're really asking for? I know that's a tough question to answer.
It's a great one, though. I mean, I have my answer for a perfect world, but we live in an imperfect world. But this is part of how I think gene therapy investigators like Barry and patient advocacy organizations like Cure Sanfilippo and many others, and sponsors, Adam, like you, and regulators. So like when you look at the whole context of how clinical trials have been done in the past, we're operating in gene therapies in a universe that's, I think, different enough. And so there have been some threads of companies actually, which may even be competing potentially fast forward into the marketplace, who've cooperated early on reviewing similar safety findings between products that would ultimately communicate or compete with each other. And that to me is very, very encouraging. And I think those types of opportunities, we have many more opportunities to do. But I think natural history data, for example, Adam, as we've dealt with in our past, one of the things that can be really frustrating is that if multiple sponsors are pursuing a rare condition and would like to have a good collection of natural history data, companies will often compete with each other in a rare or ultra rare disease for the same patients. And it can be incredibly frustrating. Whereas if everyone cooperated, again, this is utopian, but if everyone cooperated and was able to, co-fund and share data, that might work well with regulators as well, to say, okay, look, open them a little bit up to be able to speak more openly with all stakeholders, to be able to have cooperative data and to be able to do more collective analyses.
Thanks, Raf. And Cara, I think you wanted to make a point.
Yeah, I have a question around this idea of combination therapies. And so was really glad to hear Alessandro's talk and, of course, support that work, because I see that as kind of the next step as we're moving forward, and the ability to actually treat that broader spectrum of patient who may have disease burden at the time of, let's say, gene therapy or enzyme replacement or some other primary disease targeting treatment. And I just wonder, Dr. Fraldi and others, what do you see as the barriers to actually conducting combination therapy trials? Like, why are we not seeing that happening yet?
So very simple. So basically, CLEAR1 has never been tested in clinic. So this is the main challenge. So what we are doing now is trying to find support to go towards the first clinical trial. Of course, and the first indication should be Sanfilippo disease, of course. So this is what we are doing now. So just I think that our collaborator Galbitan already get some feedback from FDA and so we are. I try to find support for this. So to do the toxicology study and also, of course, the chemist, because we are also developing the drug for using the drug in clinic. So basically, we are just doing all the chemist analysis and to generate and have the drug in a large amount, of course. So this is something that we are doing in parallel with our collaborators. So the main obstacle, of course, is doing all the study towards the first clinical trial.
Thanks, Dr. Fraldi. I think, from the sponsor standpoint, or, if you're developing new drugs, you really want to understand each drug. So if there's potential combination therapy with a novel, let's say gene therapy, and a novel small molecule approach like Alessandro's working on, I think it's the challenge there is, how do you really understand each one, and how do you understand the risk benefit profile of each one first? And I think it seems like, at least in the MPS world, folks have talked about using approved gene therapies with investigational gene therapies. So you basically can use, I'm sorry if I said gene therapy, using approved enzyme replacement therapies with investigational gene therapies. For example, a model where you would try to keep substrate levels low, and if they got to a particular point, then you could come in with an approved enzyme replacement therapy. And that's a model that might be easier because you already have one approved therapy and the risk benefit profile of
and then if I time to add another important point, so about the combination therapy. So I see that the advantage of using the combination therapy is that you in principle can use, for example, low doses of vector. So you can reduce the immunogenicity and toxicity, the potential toxicity of vector, because you can have effects by combination the AB therapy with the drug. So basically the benefit should be this.
There is an example of that happening currently in about 3,500 children have been treated with Zolgensma, less than half of those in the US. But now there's also for infants, and the majority are being treated in the first few weeks of life. But now there's approval down to newborns for risdiplam. And so one of the practical considerations is, after a family might choose the single-time gene therapy treatment, if they then come back and ask their insurance company to also approve risdiplam, the costs are now significantly more, in effect doubled, and maybe even beyond double the lifetime cost of the gene therapy alone, which is $2.1 million. So that becomes a practical issue that we can make a case on appeal with prior authorization for risdiplam approval, but there have been very few studies looking at the combination, whether there's any added benefit.
That's a great point. And I know
there's likely to be similar, there's slightly different mechanisms of action, but a similar goal of increasing SMN, but it becomes a practical issue regarding insurance.
And I know we're running a little bit behind, so I think we're going to move on, unless from the panel, any other parting comments. This was really a great variety of topics discussed, all very valuable.
Yes, thank you for including.
Thank you, everybody.